It is Diagonal's Bio's turn to present here at Aktieportföljen live. And standing beside me is Jack Egelund Madsen. You are CEO. Welcome.
Yes, thank you very much for the introduction. I will get right through it.
Of course.
So I will talk about quick and accurate, cost-efficient point-of-care diagnostics. And before I start, I actually want to go to this quote from the high-impact journal Nature, which states that every single time we have an outbreak, we always one step behind because we do not have rapid diagnostics to detect a new organism. And I think that was pretty evident during the COVID-19 crisis. But this is, of course, also something that can be relevant in the future. And that is part of why I'm standing here today. Before I go into detail, I want to talk a little bit about what the problem is with diagnostic testing today. Most of diagnostic tests today are outsourced to centralized laboratories. That means long response times, delay in treatment, and also risk of spread of infections. On top of that, you need complicating and costly logistics. You need sample handling, manual sample handling, and actually talking to some of these centralized laboratories, this could actually result in up to 10% of the sample would need to be rerun. On top of that, you need costly and complicated equipment to purify the samples. There is, of course, alternatives. But in today's market, it's always a trade-off. Either you have something that's quick, but then it's slow, complicated, and expensive. Accurate, but then it's quick, then it's slow, complicated, and expensive. Or you have something that's quick, but then it's not very accurate. There have been several studies showing that the quick test, for example, have an accuracy far below 50%. In addition to this, some of the problems with today's diagnostic testing is that they're very diagnose specific. I think most of you have actually tried this during the COVID-19 pandemic. If you had respiratory problems, you would go to the test center and you would get a very binary answer. Yes, no, whether you have COVID. If this was not the right diagnosis, you would have to go back to the test center. And this also goes for other types of diagnostic tests. So what is the solution? At Diagonal Bio, we have developed new technology. We have made a new device, a universal diagnostic platform. We call it Panviral. It's unlike any other system on the market today. You see a small sketch here in the corner on the left side. It's a small portable device, has a very easy to use touchscreen, and we have a cartridge that goes in where we have 8 channels. And I'll try to highlight some of the benefits of this system. First of all, we are ultra fast using Panviral. From taking the tests to getting the final results or the diagnosis will take around 10 to 15 minutes. At the same time, we have very high accuracy. So the initial studies we have done, we have accuracy around 98 to 100%. So we are in line with the gold standard of diagnostic testing today, which is the PCR-based technologies. On top of that, we can do parallel testing on multiple samples. We can either test up to 8 patients or test 1 patient for 8 different diseases. And you can see the benefit here. Instead of having these very binary answers, we have a more holistic view of the testing. And that means that you can imagine now that we have the winter coming again. You have respiratory problems. You go up to the test center. With Panviral, we'll be able to potentially say what type of COVID you have, whether it's influenza A or B, RS virus, or respiratory bacteria. In the same way, we can use Panviral in many other disease indications. Whether this is sepsis at the hospitals, where the traditionally medical staff would treat you with broad spectrum antibiotics. With Panviral, we will be able to say within 10 to 15 minutes which specific antibiotic to use, so you can tailor the treatment very early on and get the right treatment. We can also use Panviral to test different types of sexually transmitted diseases. And again, we can test up to 8 different diseases at the same time. It's also extremely easy to use. So it has an easy-to-use touchscreen, and I will show you some of the slides later on, where we have 5 steps. Has big buttons, so anybody can basically use it. You do not need medical training. And the system is very flexible. So let's say there will be a new disease coming. As soon as we have the genetic code of that disease, we can design primers, and then within a couple of days or maybe a week, we will be able to test that on the Panviral system. The system is also extremely cost efficient because of the technology. And I'll come back to that on some of the other slides. This is an example of how testing looks with the Panviral system. In the bottom of this graph, you have negative COVID samples, so that's below the orange threshold. And on top here, you can see we have exponential amplification, and that's positive COVID samples. As you can see here, it's crystal clear and very early on. This is actually a picture from the first Panviral unit that came off the assembly line 5th of October. And here you see the same, you can say, picture. So what is the technology behind Panviral? First of all, we are using, you can say, an old technology called LAMP, loop-mediated isomeral amplification. What we are doing here essentially is that we are amplifying a genetic marker. It can be both in DNA or RNA. By coupling that to an electrochemical sensor, we can detect that very early on and very precisely. And that is exactly what we have protected with a patent here. We have protection until 2041 on the patent. So how is it actually that we differ from, let's say, the gold standard of diagnostic testing, which is the PCR-based system today? I will try to illustrate that here on this slide. First of all, with this nucleic acid amplification technologies, you need to know the genetic code. Then you need to purify the sample. Then you need to add the resources. You need to find the target, amplify the target, and then you need some way of visualizing it. With the PCR-based technology, it basically looks like this. You need to know the genetic code. You then take patient samples which are shipped to a centralized laboratory where you have robotics to purify the sample. This is a very time-consuming step and also costly. You then need to add enzymes. You need to add primers with a fluorescent tag. And then you need to go to thermal cycling, which means that you need to take the up and down in different temperature cycles. This has demands of the hardware. And then you also need hardware which have LEDs or photodiodes to actually detect the amplified marker. With Panviral, the process is much more simplified. We also need to know the genetic code, but we essentially do not need to purify the sample, which means that we save time and also costs. We still need to add enzymes. We also need to add primers, but we do not need the fluorescent tags, which is, you can say, better for the environment. And then we can do everything at a single temperature. We do not need this where we need to go up and down in specific temperature cycles. Again, saving time and costs. And then for the detection of the amplified target, we have a sensor that's dipped directly into the solution. So we do not need a device with the photodiodes or any electronics in that sense. I tried to highlight some of the benefits here on this slide. First of all, as you have all tried here, and at least to the best of my knowledge, some of the fastest tests during the COVID-19 pandemic using PCR-based technology was around 6 to 8 hours. But in most cases, this was several days. With Panviral, the whole process can be reduced to around 10 to 15 minutes. We can reduce sample preparation massively. That also means less errors, human errors. Again, we can reduce hands-on time and effort. We can reduce the electrical hardware, which also means failure by design, and also manufacturing complexity. We can eliminate optical hardware and also eliminate fluorescent and other toxic reagents, so we're also better for the environment. This is a small study that we did with samples from one of our university collaborators. So this was sampled from nasopharyngeal swabs from patients that were positive for COVID. We had 30 of these samples. If you look at in the x-axis of this figure here, then that is the cycle time, cycle number on the RT-PCR machine. So this is not minutes, but cycle number on the RT-PCR machine. On average, even with an optimized protocol, and this is actually taken from the high-impact journal Nature, the average time on the RT-PCR machine would be 105 minutes. With Panviral, the average time to detect these 30 samples as positive COVID sample was 8.6 minutes. And you can actually see the time on the y-axis. That's the time it took in minutes for Panviral to detect the same samples as we have with the RT-PCR. As you can see, there's a correlation. So we have the same sensitivity, but of course we are much, much faster with the Panviral system. And then that's one thing you need to remember here. We actually do not need to ship samples to a centralized laboratory, and we do not need to purify the sample. So we would actually be around 250, maybe 500 times faster than the RT-PCR-based technology here in this case. And that is what makes us really unique. Then you can say, why is time then that important? This is actually a study that was done between Howard and Ohio University, modified. If you look in the bottom here, then this is the control group. So if you do no testing at all, you will have 100% spread of the disease into the control group. In this case, they use COVID as an example. Then, as you can imagine, the more frequent you test, the more you can actually reduce infections. That makes total sense. So what you see here in the green bar is that if you do testing every 3 days, if it takes more than 2 hours until you get the result, you will still have up to 45% spread of the disease into the group. If you, however, can get the results in real time, so when you're actually sitting at the test facility, then you can limit the spread of infections to almost 0. And that's exactly what we can offer with Panviral. Panviral offers quick and accurate diagnosis of essentially any infectious disease. It's a universal system. We are identifying a genetic marker in the genetic material. The system doesn't care whether this is coming from Ebola, sepsis, COVID, influenza, and so forth. It's extremely easy to use the system. So the operator would be able to sit in front of the system and do all of this. So this is a one-step process. The system also allows for multiple samples to be tested at the same time, which means we can test one person for up to 8 different diseases, or 8 patients at the same time, or any combination, of course. Essentially what we're doing here is that we are taking the gold standard of diagnostic testing today and bringing it to point of care on your patient testing. This is really what we can offer with Panviral. So what are the market opportunities? Of course, depending on which market report that you're reading, it will change a little bit. It's set to around $40 billion for the infectious disease diagnostic market. That would cover everything from sepsis, COVID, monkeypox, and so forth. So how do we then plan to commercialize with Panviral? We are planning to use you can say the razor blade model. So we of course have the device, which can be used multiple times. And then we have 2 different types of consumables. We have the cartridge, this with the 8 channels here, which also contains the sensor. That's a single time use. Together with the reaction mixes, which are specific for the infectious disease or genetic marker that you want to identify. The consumables will be the primary driver of the revenue for selling the Panviral system. We're also planning to start early commercialization, and we will do that 2 ways. First of all, now that we have a research use only device ready, it's much easier for us to engage into partnership discussions with some of the big players on the market. Could be that they will in-license the technology. The second is that we will look into the opportunities to actually expand our market and segments outside the initial scope. That could be using Panviral for detection of genetic markers, or in some other areas outside the infectious disease areas for humans. So let me take you through a little bit about the history and the milestones. The company was founded in 2020, Q1, where we brought in around 10 million from private investors. We listed at Nasdaq First North Growth Market in July last year and brought in around 40 million SEK. June this year, we had our patent granted by the European Patent Office, and we are now pushing that out to the rest of the world. And that's, of course, the whole commercial foundation for actually going commercial with the Panviral system. We also did a test with 4 different viruses, just to show the robustness of the system. So here we did a test with the RS virus, influenza A and B, and COVID. And in average, it took Panviral 10 minutes to detect these samples as positive. We also signed our first letter of intent in September with Center of Diagnostics, which is located at the Danish Technical University. That is an accredited laboratory that used to test up to 12,000 COVID samples per day for the Danish government. They will do a comparison study where they will take Panviral and compare it to the PCR-based technologies but also the quick tests to give us important data on both, you can say, accuracy, costs, and time. That would be very, very interesting to follow. We also had our first research use device delivered, 1st of October, as I explained earlier on. And we are, of course, very proud that we actually got to that step with this type of speed within these 2 years. We have also done design registration of the cartridge. Essentially, what we're trying to do is to protect as much around the Panviral system as possible to protect the value. And we signed a royalty agreement with our development partner in Malmö, OIM, to make sure that we have control of our supply chain, control of our costs, and this could, of course, potentially also give us revenue in the future. And now you're probably thinking they already went a long way, so what is next? And what I can say is that we actually already started a rights issue, started 1st of December, and will continue also the 15th of December. I will encourage you to visit diagonalbio.com/investors, where you can find a teaser and more information. And just before I wrap up here, I also want to make sure that a lot of this work, gratitude, goes to the team. There's been a lot of, you can say, team effort to actually get to this point this fast. We have a lot of deep scientific knowledge within the team, also innovation, and also business development. And we are, of course, supported by a very strong and professional board. So before I wrap up here, I just want to say that our vision is to limit the spread of infectious disease worldwide by creating an accurate, easy to use, cost-efficient, quick, and universal diagnostic platform. It has been quite an amazing journey so far, and I would welcome you to follow us and take part of the journey also moving forward. So with that, I would like to say thank you, and I will be ready to take some questions.
And I'll be ready to deliver them. The Panviral can produce results in 10 minutes, and to put that into perspective, if I were to hand in tests now, I would have the results by the time I finish this Q&A.
Yes, absolutely. Yeah. So the whole idea is that you can actually get the results in real time. So basically, when you're sitting at the test facility, because as I also showed from this study between Howard and Ohio University, if you get the results very quickly, you can also limit the spread of infections a lot. So that's the whole benefit.
It's quick, but I wonder how when there's a new pandemic or new infectious disease that begin to emerge, how quick can you teach it to detect those kind of infections?
It's a little bit like the PCR-based technology, where we would need to know the genetic code. But as soon as we have the genetic code, what is needed is that we need to design primers. And that's actually a fairly simple process. Normally it will take a few days to get the primers, but of course it could also take maybe a week. But that's in that time frame.
The pandemic, of course, the COVID-19, it was a wake-up call for many, not least of all you. Diagonal Bio was founded in the midst of it in Q1 of 2020. For us here in Sweden, the pandemic and its restrictions have sort of come and went. But what sort of lessons have you taken from it?
I think the benefit, if you start there actually from Diagonal's point of view, is that everybody understands what we are talking about. Everybody have tried to take a PCR test today and understand the benefits, and everybody have tried to take a quick test and understand the limitations. And most people can actually see the benefit of actually taking the speed from the quick test with the accuracy of the PCR test and combining those two. And then on the top of that, we have this where we can also test multiple samples at the same time. So I would say it has made it very easy for us to communicate to investors and also people outside the company and make them sure that they understand what we are talking about.
But we're heading into 2023 and Q1 of 2023, and that means you've been a company for 3 years, and already you have a product. What sort of driving factors have ensured that you've been able to accomplish that?
I think you can say that we have been pushing really fast, push hard every day. We have a very strong team who has been dedicated to actually deliver. And of course, you can say also the money that we received from listing at Nasdaq First North have also made it possible for us to actually accelerate the development.
And now that you're taking in new money, are you wishing to further accelerate this development?
We are of course pushing very hard to make sure we get to a stage where we can have a commercial system. And that's of course the goal of this, to make it a successful business.
Something that will help you commercialize is the pilot studies, and you've signed one letter of intent to begin those studies at a Danish university of technology, that is. Why are they the right partner for testing the product?
You can say Center of Diagnostics is an accredited laboratory, and they have actually, during the COVID-19 crisis, they tested up to 12,000 COVID samples per day for the Danish government. So you can say they know exactly what it is that's needed, and can also see a clear benefit of actually using a system like ours. So of course, getting this data out where we can get a comparison to the existing technologies compared to Panviral would be essential for us as well, I think. But we can of course also look into other disease areas and other partners. But this is, you can say, the first important step that we take.
When can we expect the first feedback from these studies?
This is always this with the timeline and coming with the commitments during the investor talks here. What I can say is that we signed a letter of intent. We do have the research use only instrument out. And we are pushing very hard to get the last details in place so we can actually start these studies. And I think I'll leave it with that.
No concrete timeline then. But for investors that are curious about how this develops, how will you communicate? How will you communicate updates on the study?
We will of course make sure that we communicate. So the second that we start the study, we will communicate that out so people know that we have started the process. And of course, then we will also come out with communication when we have finalized the study as well.
So in your current heading, as you explained before, you're heading towards commercialization, and it is the key here. For 2022, you've created a checklist for 2023 as well, and one item states as follows: identify suitable regulatory alternatives for use of medical diagnostics in global markets outside Europe. Have you identified any alternatives thus far?
We have looked into the process in the US. And you can say under the FDA's there is what's called an emergency use approval. And that's actually meaning that you could actually go quicker to market because if the FDA have a list, and I think they have between 10 and 12 infectious diseases on that list, that's really deadly. And if you can test those diseases, you can say that you can get permission to go onto the market because the cost benefit actually permits that, even before you have the full regulatory package available. So that's of course something that we are looking into.
Are there any diseases in mind that you want the product to test?
Right now, you can say COVID is on that list. In many of the European countries, they have downgraded that and taken COVID off as a deadly disease. But right now, that's still on the list in the US. But there are other viruses like Zika virus, Ebola, and things like that we can also test with the Panviral system.
And I assume you have the genetical code for those?
I think that's old viruses, so they should be sequenced, yes.
And if we focus back on that checklist that you've made towards commercialization, for 2023 you want to initiate the process of regulatory approvals. Do you specifically have a goal for when you're supposed to initiate this in 2023?
It's always again this with the specific promises. I don't want to go into and be too specific, but of course we want to initiate this so we can get the product, you can say, final, also so we can actually start selling it for in vitro diagnostics. But I will not give you any concrete deadlines on that.
Of course. Jack, that was all of my questions, but I thank you very much for your time, for being here presenting and answering my questions, and I'll wish both you and Diagonal Bio continued luck in the future.
Thank you very much. Pleasure. Thank you.